let me talk a little bit about the Nobel Prize. I don't know if you've thought much about it. You're all young enough to strive for getting one, because if you look around when people get Nobel Prizes, they're pretty old. So you have lots of time, and you can try to get one. And the fact that this time, at least in physiology and medicine, I believe it's the first time that the Nobel Prize in Physiology and Medicine has been given to an individual that had done the entire work in a company. I have looked at many of the 222 prizes, and it's really difficult to find the detail, but I didn't find a single other prize that has been given to an individual in physiology and medicine for the work that he or she did at a corporation. So Michael Houghton did his entire work at the Chiron Corporation. Which at the time was a very important and very successful California biotech company. Bit more statistics, the chemistry prizes didn't look all that much better. Two prizes out of 185 were given to an individual in a company, and only the physicists did a little better. 19 out of 215 physics prizes were given to individuals that did their work in the company. So let me tell you a little bit about hepatitis C, or what became hepatitis C. So it all started with actually Harvey Alter, who also got the Nobel Prize. He worked at the NIH then, and he still works at the NIH, and he's even older than Peter. Harvey was the one that did methodical studies of what we call then transfusion-associated hepatitis. There had been testing for hepatitis B and hepatitis A. Those tests were available for quite a number of years already, but there was still hepatitis that was apparently transfusion associated, and Harvey led the group that showed it was indeed transfusion associated, and his team was responsible for calling it non-A, non-B hepatitis. So after he had defined non-A, non-B hepatitis, people tried to find an additional virus. Traditional techniques for finding that virus were tried. It's isolating the virus and cultivating it, and see if you can find it. People tried that for for more than a decade, and failed. So Michael Houghton was the first one, and he and his team said, we can't beat a dead horse. We are going to use a new strategy, and we are going to isolate the genome of the new virus, and that's what later became, or shortly after they succeeded, became hepatitis C. Isolating in genome out of blood, and it was out of chimpanzee blood, was certainly not embraced by everybody. They got a lot of criticism. I was not part of this time. I really know nothing about it, but just like you, and I can read online, there was a lot of criticism. How long did it take? I can't tell you anything about what happened within Chiron, but I know that Michael began working around the approximately end of 1983, and he and his team in October of 1989, so nearly 7 years later, they had the two famous science publications. So the two publications came out in one issue of Science back to back. And I'm going to read the titles. So the first one was isolation of a cDNA clone derived from a blood-borne non-A, non-B viral hepatitis genome. And Michael Houghton was last author. And the second paper was called An Assay for Circulating Antibodies to a Major Etiologic Virus of Human Non-A, Non-B Hepatitis. So that second paper, where Michael Houghton was also last author, also had Harvey Alter as an author, because it was Harvey who had given the samples from the infected, from the blood people that had been infected by blood transfusions, and his sample bank made it possible to connect the genomic finding of the virus to circulating antibodies in humans. So what? Why did I say Michael Houghton and his team all the time? You might not have noticed it, but I was always trying to say Michael Houghton and his team. I know Michael well enough to know that he was extremely conscious about his team. The work he did was far too gigantic from one person and a technician. It was a large number of people that all pulled in the same direction. And Michael actually refused to accept the Gairdner Prize, which is a pretty substantial Canadian prize, only a few years ago, and I quote, because two close collaborators were not being similarly recognized. That was his reason for refusing the prize. And these two collaborators were Quilin Chu and George Kuo, who worked with Michael at Chiron when they cloned and identified the hepatitis C virus. And it was George Kuo who was first author on the paper that I just quoted, the assay development and finding circulating antibodies. George Kuo, in fact, was the person who developed the antibody assay that was used to prove the virus connection to non-A, non-B hepatitis. And I worked closely with George and used his initial work to develop the first hepatitis C antibody test at an industrial scale. And just to say a few words about the impact of this work, not my work. In the end, somebody had to go from the lab to a big scale, but what was the ultimate impact? I am going to quote and read to you, according to a recently published statistic, it is believed that Houghton's team's discovery has led to screening tests that have reduced the risk of acquiring HCV through blood transfusions from 1 in 3 to about 1 in 2 million. Think about that for a moment. 1 in 3 down to 1 in 2 million. If that isn't impact, I don't know what impact is. Give you another statistic where it is estimated that antibody testing, which is done still today at every on every blood that is ever used in medicine, antibody testing is preventing at least 40,000 new infections per year just in the US. So I've told you that it wasn't me who developed the first antibody test, it was George Kuhl. I simply made it big. And so in order to give you a glimpse into what I did and what you might someday experience if you go into commercial science and product development, and so I'm going to have to bore you for 1 minute with my background, which will explain what I did and why we could do it in such incredibly fast time. So I have a degree in medical microbiology, and after my PhD, I joined a German diagnostics company. The older ones of you, not fellows, but faculty, may remember Behring Diagnostics, the company that has its name and was co-founded by Emil von Behring, one of the early Nobel Prize winners. No, he was actually the first Nobel Prize winner for medicine, but he didn't work at Behring at the time. So he still worked in academia. So I joined this company. It happened to be in the town Marburg, where I did my PhD. So it was convenient that I got a job there. And it was a rather boring job. The company had a hepatitis B assay and a hepatitis A assay that was successfully sold into blood banks and blood screening environments. And I was supposed to collaborate with the engineering group to put it onto a semi-automated testing machine. Now, can you imagine working with engineers? They always say, no, this doesn't work. Can't do it that way. So there I was. It was actually a very rewarding experience. It broadened my skills very early on, and in the end, I didn't find it a boring job. I was thrilled to have gotten the job, but I didn't know what I was going to get into. And I also had to realize when I looked at these blood screening laboratories, I mean, testing machine was essential. I mean, it's a very high throughput environment. I had no idea. But then I got lucky. My medical microbiology degree was wanted again because the HIV virus was discovered. And HIV was obviously the big concern, not obviously, but it was at the time the biggest concern for the safety of the blood supply. And Behring did have a lot of products that were derived from human blood, and they were using it in coagulation pharmaceuticals. And they are still very successfully used in that environment. Similar products today, different companies, different names, but they hail from originally Behring. And so an HIV test was of critical importance. And I was put in charge of the team to get it developed, put on that dreaded machine, that would run them fast and get regulatory approval. And I am proud to say that I was the first one this was the first test that gained regulatory HIV test that gained regulatory approval in the world. But again, regulatory approval only in Germany, and who, after all, cares about Germany? So Abbott Laboratories, have heard that name recently? They were an international diagnostics powerhouse then, just as they are now, and they were next to get an HIV test approved. And as you may imagine, today everybody believes that they were first. So this is my background, and this is why after I went to the US in 1987, I was offered a job at Johnson & Johnson to help them with their ailing blood screening business. I got another boring job. Hepatitis B test improvement, HIV test improvement, hepatitis A test improvement. But I got my green card. So I was only too happy to work on these improvements. But then I got lucky again. As I had worked for nearly 8 years in the blood screening business, or what did we call it? The corporate lingo wanted it to be called the safety of the blood supply. So since I was there 8 years, I was aware of non-A, non-B hepatitis, which had already been defined, and Harvey Alter was known to everybody who worked in this industry as the person who had defined it. And it was known that it infected tens of thousands of, or it affected tens of thousands of people by blood transfusion every year. So, so in the 8 years I worked in industry till 1987, I had heard from probably 10 groups, if not more, that they had found the non-A, non-B hepatitis culprit. It never turned out to be the case. So here I was in 1989, and there was hepatitis, non-A, non-B hepatitis again. Now, a very quick tidbit, my employer, J&J, had made a very significant equity investment into Chiron Corporation in the mid-80s. Chiron Corporation, you recognize Michael Houghton's employer here. So in the fall of 1989, a week after the two Science articles came out, I was told that non-A, non-B hepatitis virus had been discovered at Chiron, and I better get into the plane with the J&J executive team and learn what happened at Chiron and get an antibody test developed for our blood bank business as fast as I possibly could. I do admit that I was ultra skeptical when I boarded the plane to San Francisco because, as I just told you, I had heard this so many times. But I did enjoy the first class flight. And without the executive team, I would have been in the back of the plane. And then I met first George Kuhl. Remember his name? The team member that did the first circulating antibody test, and I met Michael Houghton and many others at Chiron, and Harvey Alter at the NIH. And I think it took me approximately 5 minutes to be convinced that this was real. So now, what is remarkable? What did I do that I would consider remarkable? I didn't do it alone, but it happened, and I love to share that with you, and then I shut up and let you get to the fun part. So what is remarkable is the timeline. The Science article was published in October 1989, remember? The work in my laboratory at J&J to take the basic information from George Kuhl and develop an assay that could be manufactured in high volume took less than 4 months. In March of 1989, we had manufactured 3 lots of assays with 10,000 tests per lot. This is an FDA requirement. We had to do this for FDA approval. It was not because J&J would ever do anything these tests. They were only used to complete the clinical trials at three independent sites, another FDA requirement, and this was done within 1 month. And the test, month of April, we're now in April, and in mid-May, the test was approved. Unlike today with COVID, there was no shortcuts, no emergency approvals. I don't even know that emergency approvals existed in those days. It was made happen by a very dedicated team at Chiron, at J&J, at the NIH, at the CDC, and at the FDA, and all were working together. We had meetings with the FDA team on an almost daily basis in April and May and until we got it done. And you picked up the phone at the FDA and the leader of the team answered the phone. So I have to admit, there was lots of money too, and that helped. We didn't have to worry about how are we going to pay for this, how are we going to pay for that. J&J had all the money that we needed to make this happen. But money isn't everything. Now I'm telling you why I'm saying that. It turned out that the FDA required that there would have to be a diagnostic test and not just a blood donation screening test. Now you may ask, what is the difference? I tell you, there is no difference. It's exactly the same test, technically. But from a business perspective, it's a completely different test. And J&J had no presence in the diagnostics business. So diagnostics business, meaning you go to the doctor, you have symptoms, you get a hepatitis test, and the doctor says, we're sorry, but you have hepatitis and you need you have been infected with hepatitis C. You need to do this, that, and the other. So I really was naive in those days, and I said, that can't be that big a problem. With the money of J&J, they can buy a diagnostics business. And they could have. They did much later. They bought Kodak's diagnostic business for billions and billions of dollars about 15 years later. But in those days, when that hepatitis C question came up from the FDA, let me say it, some executive ego got in the way. No detail here. But we wound up licensing the diagnostics rights to the big enemy. Guess, Abbott Laboratories. Remember the name? So I was closely involved in the licensing negotiations, but I'm just going to give you one tidbit from it that is amazing and requires to be mentioned. So Chiron, J&J, and J&J jointly negotiated with Abbott the highest royalty rate, the second highest royalty rate that was ever paid in the diagnostics industry. More importantly, the royalty was negotiated not as a percent of revenue, as it is customary, and you probably have heard about percentage royalties, but it was negotiated to be an absolute dollar amount per test shipped. So something, when you have nothing better to do, you can think about the difference. Why is that so much better? I'll tell you why it's so much better. It doesn't matter it didn't matter how Abbott was going to ship, if they were giving tests away, if they sent them away at a very low price, a percentage of revenue, you go as the recipient of the royalty, you go up and down with the decisions of the licensee. But if you have an absolute amount per test shipped, and an FDA regulated test has to have reviewable shipping records. So there is very little you can't cook those books. And so you are in control of pricing indirectly. It's a very harsh deal from a capitalist perspective, a very good deal from a capitalist perspective, a very harsh deal from a patient perspective, probably. But that's what it was. And I think it's just an interesting tidbit. And now I let you go to the fun part. Only last thing I want to tell is that all of this happened while my first company had already gotten funded, and my then colleague and best friend, now husband, Peter Hansen, was working in our laboratory to start our first company. But I didn't think it was ethical that I leave this project in the middle of this activity. So I only left after the test was licensed and the first shipments, FDA approved, and the first shipments had gone out. So now you know the Hep C story, and think about what you will do to get the Nobel Prize, or maybe participate in some way in the next Nobel Prize.
Thank you so much for sharing the story, Petra.
That was very nice.
So we are incredibly lucky and incredibly honored that Michael himself agreed to tell you a little bit about what happened.
Thank you very much, Petra. Great to see you after all these years. Brings back a lot of memories, good memories.
It does.
So yeah, very nice to be talking with you all. As Petra mentioned, the discovery of HCV was a team effort. This is a photograph that's way out of date, but it was published in a Nature Medicine article in 2000. And I look completely different to the person in the top left, but gives me an opportunity to acknowledge the roles of Dr. Chu, who's top right. He hasn't changed much, actually. Dr. George Kuo, bottom left. So these are the two people I work with very closely, which succeeded in discovering HCV. And then also, we needed a collaborator in the animal transmission field. There were 4 or 5 labs around the world that had transmitted non-A, non-B hepatitis, as it was then known, to chimpanzees. And we needed fresh material. We started off working with human materials from the Japanese NIH, but these were autopsied materials. And when we extracted the nucleic acid, the RNA was highly degraded. So it was important to work with a animal transmission expert, and that was Dan Bradley of the CDC, shown in the bottom right. So yeah, team efforts are usually required, and certainly was in this case. So before I actually show you the one experiment that worked in 7 years, let me just give you a little bit of background. Non-A, non-B hepatitis was identified in 1974, 1975. Hepatitis A and hepatitis B had been discovered. Baruch Blumberg discovered Hep B in the 60s. He got a Nobel Prize for that. And then hepatitis A was discovered by Bob Purcell and Steve Feinstein at the NIH around 1973, I believe. Blood tests were developed to both of those viruses, and everyone was assuming that the transfusion-associated hepatitis that occurred was due to either A or B. But it turned out not to be the case. Most cases of transfusion hepatitis turned out to be neither hepatitis A or hepatitis B, or CMV or EBV. Occasionally, herpes viruses can cause hepatitis following transfusion. And so hence the term non-A non-B hepatitis was used. And it took, almost 15 years before we published our work to actually identify the causative agent, which turned out to be hepatitis C. So all the procedures that were used successfully to identify hepatitis A and hepatitis B, those were applied, obviously, to non-A, non-B hepatitis. They didn't work. We know now that they didn't work because the titer of virus in Hep C is much lower than in Hep A and Hep B. So the techniques used then just were not sensitive enough. So I got involved. I was trained in England, and I went to Chiron, this startup biotech company founded by two professors from University of California, Bill Rutter at University of California, San Francisco, and Ed Penhote at Berkeley. They set up what was then known as a genetic engineering company, trying to use recombinant DNA methods and associated technologies to do something good in healthcare. So I moved there in '82 as a senior scientist. I had my own lab. I was going there to work on interferons because that's what I had been working on in England, human interferon genes. I was very interested in making artificial interferons through recombinant DNA, chimeric interferons, but also type 1 and type 2 interferons were known at that time, but there was a growing swell of information suggesting there was another class of interferons that had yet to be discovered. So I was kind of interested in that, so-called epithelial interferons. Turns out they have been discovered now, and they are known as type 3 interferons that work especially at the mucosa when pathogens like COVID enter. These work primarily at the mucosal level. Anyway, after discussions with Dino Dina, who was director of virology at this company called Chiron in California, and with Bill and Ed, I decided to try to identify the causative agent of non-A, non-B. The reason for that is many of the techniques that I was familiar with in England, looking at human interferon genes, could be applied potentially to discover non-A, non-B. So we started off with the human materials, and after 9 months or so, one of a good friend of mine, Tatsuo Miyamura, he came over to work with me on this issue. He brought human samples from cadavers, and as I mentioned earlier, the quality of the RNA was poor, and we felt this would not be a good approach using molecular biological methods. So we needed to find a collaborator on chimpanzee transmission, and we went to Phoenix. Bill Rutter and I went to Phoenix, Arizona, to the CDC there to talk to James Maynard and Dan Bradley on whether they would be interested to collaborate with us. They were, and Dan Bradley provided us with a ceaseless supply of materials from his animals. So what he had shown, along with other groups, that you could transmit non-A, non-B hepatitis from human samples to chimpanzees. You could passage it in chimpanzees, and it looked like the disease was similar to humans. So at that point, we started off cloning all the mRNA in Dan Bradley's livers, his chimpanzee livers. And the question that posed in front of us was, how do you figure out which ones are from the virus as opposed to the chimpanzee genome? So there's a method you may be familiar with. It's an old method. It's called plus-minus hybridization. So to find out if a clone really could be from the virus, you make cDNA from infectious livers. You make it highly radioactive. I used to use 10 millicuries of P32 about every 2 weeks. So it's amazing that my fingers are not glowing in the dark. I did that for several years. You also make highly radioactive cDNA from uninfected livers. You hybridize both probes to the cDNA clones, obviously separately. And you're looking for a clone that hybridizes with the probe from the infected liver, but not from the control. So it's really a laborious, very labor-intensive job. So after a few years doing that, we came up with a lot of clones that seemed to be specific to the non-A, non-B infected livers, but we were not able to show that any of them were derived from the virus. At that point, we went back to Dr. Bradley and said, look, we need to improve the materials we're getting from you. Not a criticism, but just a practical point of view. Can you give us high titer materials? Now, high titer is still low titer compared with Hep A and Hep B, but we were aware that groups at the NIH and in Japan and at the New York Blood Center had chimpanzee and human materials that had been shown to be relatively high titer. And you know, I'm almost ashamed to say this, but the way that the titer was done, the infectious titer, we didn't have any cell culture. We had no marker for the virus. So all that people could do is actually dilute samples and inject them into naive chimpanzees. And so in that way, various groups around the world had shown they had sera of at least 1 million chimp infectious doses per mil. Our materials coming from Dan Bradley had not been titered. And so we thought, well, look, we can't keep searching for a needle in a haystack without knowing that we've got materials as good as everyone else. So we asked him to go back and titer his samples. So he made pools based on his data and titered them. And then we got a plasma from chimpanzee 610 that had a titer of around 1 million infectious doses per mil. We applied our plus-minus methods to that, still no success, still couldn't find the virus. In the meanwhile, we were trying lots of other methods. There was evidence from around the world that maybe non-A, non-B was related to Hep B. Maybe it was related to picornaviruses, retroviruses. Some people were postulating it was a prion. So what we did is we used hybridization probes from other viruses that people thought was related to non-A, non-B. No success. We failed to see any cross-hybridization. And we now know that's because Hep C is quite a unique virus. Sequence-wise, it's classified within the Flaviviridae family, but it's a very diverged genus within the flaviviruses. We use yellow fever and dengue as hybridization probes. Bovine viral diarrhea virus, which is the toga virus, as a probe, nothing. And Hep C is classified within a Flaviviridae family, but very distant from the togas and the flavies. The NIH also came up with some information that maybe Hep C, non-A, non-B, was related to hepatitis D virus. So my lab, we characterized the hepatitis D genome for the first time. showed it was a very unusual viral genome. It was actually related to infectious agents seen in plants, viruses and virusoids, circular, single-stranded circular RNA genomes. So the Delta virus is a very interesting virus that's emerged in humans. But unfortunately, once again, having characterized that, we were unable to use it as a hybridization probe for non-A/non-B. We got no signals when we did that. So that was the history of searching for this virus. It wasn't easy because it was done in a biotech company, obviously, that had investors, and investors want to see return on their money quite quickly. In those days, they were a little bit more lenient than investors today, who generally investors want to see a 3-fold return on their investment or a 5-fold within 3 to 5 years. So it was fairly stressful doing this project within a commercial entity. But on the other hand, to do this in an academic setting would probably have been very difficult, because you can't put students and PhD and postdocs onto a project like this where there's a high risk of actually getting nothing back, and their careers would be ended. So somehow we need to find a way where serious medical problems and diseases like Hep C can be worked on by academics as well as industry. So it's something I would like to work towards helping. Anyway, look, that was
talk about that. We should talk about that.
Yes.
I couldn't agree with you more.
Yeah. Let's come back to that then. I'll just be just now a few minutes more on my slides. So I just wanted to show you the one experiment that worked, and it's schematized on this slide. So what we did was we took Dan Bradley's chimpanzee plasma in the top left, and we assumed it was a virus, and we assumed that it was the smallest known virus in terms of size. And so we ultracentrifuged such that the smallest known virus to man would be pelleted. We didn't assume what kind of virus it was, whether it was an RNA virus or a DNA virus. So we extracted total nucleic acid, and then you use reverse transcriptase that copies not just RNA, but also copies DNA into a complementary copy. And then we cloned it into an expression vector, lambda GT11. This was developed by two scientists at Stanford, and they used GT11 to identify the T-cell receptor. Dr. Davis and forgotten his colleague's name now, but anyway, it was two people that identified the T-cell receptor using lambda GT11. So it was a cloning vector, but importantly, it expressed the cloned nucleic acid into protein. You could induce expression. So essentially what we ended up with was a huge proteomics library. This was before proteomics really dawned, but essentially that's what it was. It was millions and millions of clones, each expressing a protein from a piece of nucleic acid from this plasma. Now, non-A/non-B patients, we knew that most of them had persistent liver disease. It was unusual for them to recover spontaneously. And nobody had identified antibodies to a etiologic agent of non-A/non-B. So, but nonetheless, we assumed that there were antibodies circulating in these patients to the virus. And so we put the two together. We put the proteomics library together with the antibodies, and we were looking for positive interactions. Interestingly, in order to clone this minute amount of nucleic acid from the pellet of the plasma, I had to use a carrier. So I used the bacteriophage RNA, MS2 bacteriophage RNA, very tiny quantities, but if you don't use a carrier, you'll lose the Hep C RNA. And by the way, I should have mentioned, this is all before PCR. This was before PCR was invented by Kerry Mullis, who actually worked in the company next to ours at Cedars. and CDUS eventually was absorbed by Chiron. So this was before PCR. It was before you had the ability to sequence genes quickly. It typically took around a month to sequence about 100 or 200 nucleotides. A couple of the positives turned out to be as a result of antibodies in non-A1B patients that had antibodies to bacteriophage, MS2. So it's a very powerful technique. But eventually we got one clone, 511, called 511 by my colleague, Dr. Chu. I think it was, plate number 5 and autoradiograph number 1 and so forth. And then we had the problem of, well, how do you prove it's derived from truly the etiologic agent of non-A1B? We proved it by showing that 511, which was only very small, it was only about 100 base pairs, it was not derived from the chimpanzee or human genome. We used southern blots to do that, very old technique. We showed it was derived from an RNA molecule that was very large, about 10,000 nucleotides. You only found that RNA in non-A1B infected materials. And then crucially, we developed an immunoassay for the protein encoded by 511. And we saw that patients diagnosed with non-A1B had antibodies to it. And also Dr. Bradley provided us with samples from chimps that he had infected either with non-A1B or Hep A or Hep B. And we only saw seroconversion to 511 in the non-A1B infected samples. So that convinced George, Quilim, and myself that we had identified Hep C. We actually filed a patent at the end of 1987, and we called it Hep C. And the work thereafter was really involved in proving that this was the major cause of blood-borne non-A1B hepatitis, and that is the case. This slide just shows with our first generation assay that when we looked at transfusion-associated non-A1B in the United States, 71% were positive, whereas in normal controls, it was 1 to 2%. The next slide shows when we got samples from around the world, for example, from Dr. Benino in Italy and Dr. Miyamura from Japan, we saw that chronic non-A1B hepatitis cases had antibodies to 511 at around 80%. Given that 511 is a very small antigen, this was, what it shows is that 511 was a very immunodominant epitope of Hep C. So I think probably I've said enough. This is a scheme of the genome of HCV. 511 came from what we now know is part of non-structural protein 4A. So it's not a virion protein, a structural virion protein. It's involved, NS4A is involved in the replication cycle. It's a very interesting protein actually, but it contains a very strong B-cell epitope. So as Petra knows, what we did at Chiron, working with our partners, Ortho Diagnostics and J&J, is we slowly walked the genome to identify the entire sequence. We did epitope mapping experiments. We found lots of B-cell epitopes all over the genome, and that resulted in a series of diagnostic tests. I think we had at least 3 successive diagnostic tests, each containing more epitopes, more sensitivity with high specificity. And then, of course, I should mention that the 5' untranslated region of HCV, right up here, is highly conserved amongst all the diverse HCV strains. And so PCR assays and TMA assays could be developed based on this 5' untranslated region. And of course, right now, the blood banks around the world use antibody screening as well as nucleic acid screening. Well, there's more to the story, but I'm going to end there and happy to take questions. We can come back to that question of how do we get attention on major diseases which academically would not be, shall we say, profitable for students and postdocs. And in industry, it's difficult to work on them because you don't know if it's going to draw blanks after spending a lot of money. Maybe you have some ideas on how we can do that, because I think it's something we have to do. I'm very conscious that today, with all of our sophistication, if you want to use that word, we have so many diseases where we have very little knowledge of what's going on and very little way to treat them. You know, Alzheimer's is a great example. Inflammatory bowel disease, multiple sclerosis. Of course, cancer gets a lot of attention because if you can prolong someone's life by 6 months, that translates to benefit to patients and also to a lot of money, to be frank, for the companies. But other diseases are neglected, and somehow we've got to get more attention on them. Any comments on that, on what people think we could solve that issue?
I'd like to invite some of my academic colleagues, as you all know, and Michael, you know as well. I have spent all of my life as an industrial scientist, other than a very brief period in the beginning. I have joined MIT as a teaching faculty a number of years ago, and I'd like Martha Gray, who is actually the leader of this project, and I believe she is with us. There she is. Yeah. She created this teaching project where we are trying. Martha, you tell what you created.
Well, first, Michael, thank you so much for joining us today. I'm sure Petra told you today's a very special day for this group because they've just finished what we call phase 1 of the Catalyst project. And so it's just an honor. And Petra has been a core lead faculty in this edition. So, you know, the problem you point out is one that's near and dear to my heart. And in fact, in a sense, was the impetus for creating Catalyst to begin with. The way I usually phrase it is that people go into biomedical research, in my experience, usually because they want to have an impact on patients in some way. Whether they're the ones that carry through every step is unlikely and not necessary, but they want their work to ultimately have that. And yet it almost never happens. And so our hypothesis is some of that has to do with how do you think about the problems you're going to solve and where do you focus your strategy in going forward with it, which does not end with the paper, as you know, right? The impact you had wasn't because you published it, it's because of all the stuff that happened, right? And you didn't do it alone. So it was like we wanted to break those bounds that it's all about one person, it's all about getting the paper. Not that papers are bad. I've been accused of not liking papers. Of course we need them, but it's like, okay, what stands between where you are now and actually having the impact? And that forces you to reckon with things that might be pretty risky. So it's not whether or not it's risky or not, it's whether or not, you know, as long as you're not being an idiot, right? That if this works and there's plot, it doesn't violate the laws of physics, right? You don't need a time machine, right? And if it works, then it has a huge impact, which, you know, I think your story illustrates brilliantly. Your story also illustrates the fact that technology can be an incredible enabler. Because like like you were saying, if you had to do it now, it would be a lot different. And so our programs you'll see in this room are engineers, scientists. Petra has taught me commercial science. It took her a while for me to understand. she'll acknowledge I finally had that aha moment that when I called her a business person, she used to kill me. So at any rate, I've been transformed. But I think all of these different perspectives coming together to move projects along, or both to nucleate new projects and to then strategically think. So that's what we're trying to do in fits and starts. And we Actually, it's not about us faculty, it's about all the fellows that jump in, because they're doing just what you say is hard. They're saying, I'm going to not do things the usual way all of my colleagues are doing it in their university. I'm going to step out of that, and I'm going to take the risk to spend my time when I don't know for sure that I'm going to get some academic bean at the end in a year or 2 years. So they're the superstars.
Maybe it's a bit like handling a financial portfolio, you know, your personal portfolio. You don't want to put it all into one company or one investment. You want to share it out just in case. And, you know, maybe, you know, having been at the University of Alberta for 10 years now, I work with a lot of really excellent faculty, very specialists in their own field. They tend to use model systems, like, you know, for example, a herpes virologist will not be looking to see if herpes 6 is causing a disease in humans. You know, there have been linkages between various herpes viruses and human disease. They will use a mouse herpes virus to teach students about the life cycle and how to operate in molecular virology, how to learn the technology. And I think maybe, you know, I'm going to be talking to student groups in as well as yours. And I think what I'm going to recommend is, look, 20% of your time, devote it to a difficult area of unmet medical need. You do need to get your papers. You do need to get your career started. But the other thing, I think, is we need the funding agencies like NIH should be funding much more speculative research than what they do. And in Canada, it's a great country, and I've really enjoyed working there for 10 years. A lot of good scientists, but what they call the CIHR, which is the equivalent to NIH, they tolerate virtually no risk in a researcher's proposal. It's all got to be mapped out with, so I think
NIH.
we need changes in the funding agencies to do this.
Totally agree with you. That the reason people do the work the way they do is because that's how it's funded, that's how it's rewarded. You know, that, so I get it.
And obviously we do need basic research, absolutely.
Absolutely.
We can't do it without basic research discoveries. So yeah, it's frustrating to me, I have to say, that, you know, when I started my career almost 50 years ago, we knew about multiple sclerosis. We knew about Alzheimer's. We knew about inflammatory bowel disease. And then 50 years later, we know hardly anything new.
I started out in orthopedics the same way.
One of our fellows got really passionate about a multiple sclerosis project, and he will probably agree with you.
Interesting. Well done.
I think one of the things that we learned to do.
David, who just showed his face.
I like very much the use of that phrase, unmet medical need, because that's the beginning of our course. When we run this course through different cohorts, it starts by, how do you identify an unmet medical need? And it's not easy. There's a lot of validation required for that need. How do you go talk to stakeholders in these issues, and how do you sift through the information to find out if is this just your dream project, or is it really a need? And it's hard to do. Go out and make cold calls, and how do you have a conversation with someone you've never met who has an important idea to share with you? And this program seems to have some very strong success in that first step, which I'm very pleased about.
Yes, excellent. Congratulations to the faculty and all the students. Congratulations on your graduation today.
Well, we decided to keep the congratulations screens up for you, because you deserve our most heartfelt congratulations for finally having gotten the reward you probably deserved a long time ago.
Yes, I had given up on the Nobel, frankly, and it was never a strong goal of mine. You don't, as you said earlier, you don't get into this area to get
Absolutely
awards.
not.
We're all trying to improve, to
Absolutely.
do our
But
bit
I
for
did,
humans.
when I spoke a little earlier about my little part, I did tell the fellows they were young enough to still think about the goal of a Nobel Prize and look around how old Nobel Prize winners are. So you have a long time to get prepared and work hard, and it just might happen.
Yes. And,
Michael, we thank you so much for taking the time. I think I can speak for all the fellows that we're extremely honored that you took the time.
it's been a pleasure, and I think your students, you've got so many technologies now to apply, and they'll be growing exponentially, and you have so much opportunity to try and impact major diseases. And we want to see we need more female Nobel Prize winners. So the ladies in the audience, go for it. As well as the men.
Go ladies. Thank you again.
Thank you very much.
Thank you very much.
Thank you.
You're very welcome. Good luck, everyone.
Okay.
Thank
Good luck.
you.